In this video, I'm going to discuss two questions on the problem set that concern the energetics of interactions between bases in double-stranded DNA. And so these are questions 4 and 10, which deal with the relative importance of the so-called base stacking interactions and hydrogen bonding between bases. And the first question asks, why is it that hydrogen bonding contributes less to the net stabilization of the helix, that's the DNA double helix, than base stacking? And the other question simply asks, why is base stacking important? So to begin, let's look at some Watson-Crick base pairs in DNA, shown in this diagram. And in the canonical Watson-Crick base pairs in double helical DNA, shown on the left, and in RNA, shown on the right, you can see that there's a set of hydrogen bonds indicated by dotted lines. I'll denote that by H bond between the edges of the base pairs on one strand and the bases on one strand and the bases on the other strand. The only difference in terms of the hydrogen bonding between RNA and DNA is that thymine is replaced by uracil, which is very chemically similar. The hydrogen bonding comes about because the bases are polarized with small amounts of negative charge and positive charge in individual atoms. For example, you can think of the oxygen as slightly negatively charged, which we write as a delta minus, so that's not a full charge, it's a partial or fractional charge. And the hydrogen is a fractional positive charge, and the nitrogen is again a negative charge. And so these electrostatic interactions, including the partial positive charge on the carbon, give you dipole-dipole interactions between the groups, which are effectively what the hydrogen bonds are. So in a double helical DNA or double helical RNA, the geometry of the base pairing is such that A can form 2 hydrogen bonds with T, and G can form 3 hydrogen bonds with C. And this pattern of hydrogen bonding is one of the components of the ability of the replication machinery in cells to replicate DNA with high accuracy. So let's now look at what base pairing is. Base stacking is. So in this diagram, you can see an edge-on view of B-form DNA, which is one of the configurations of DNA, the most common one. And what you can see is that each base pair, so each one of these things is 2 bases, and we call that a base pair, is stacked on top of the next base pair. So this stacking of parallel base pairs is what we call the stacking interaction. As in all interatomic interactions, there's a very small van der Waals attraction that underlies all interactions between one atom and another, and they add up over here to give you a slight net attraction. But the most important component of the base stacking interaction is not the van der Waals attraction, but is electrostatic complementarity. And I'll explain what that is in a minute. But before I do that, let me just say that the van der Waals interaction between atoms has a very important consequence, which is that the spacing between adjacent base pairs is ultimately determined by the van der Waals contact distance. That's the closest approach between carbon, nitrogen, and oxygen atoms, beyond which the van der Waals repulsive turn kicks in and the energy goes up very sharply. So this fundamental spacing between base pairs of 3.4 angstroms along the vertical direction between base pairs comes from the van der Waals repulsion. So the most important aspect of van der Waals energy and the interaction between bases is determining the distance between bases. The attractive component is there, but is not so strong. So how does this electrostatic complementarity between bases manifest itself? And it's also a consequence of the polarization of the bases that leads to the hydrogen bonds along the edges. Now, if you look at the 4 bases, A, G, C, T, as shown in this diagram, they are all slightly polarized so that the oxygen atom has a net negative charge, again denoted delta minus, and that's shown in the diagram below with a red color. So we can, what this red color here means is excess partial negative charge. And likewise, the nitrogen atoms tend to be blue, and which is because they have the hydrogens in particular on the edges have a net positive charge. So the regions colored blue are regions of excess positive charge. Now this calculation, this color-coded diagram comes from quantum mechanical calculations of the electronic distribution for each of the bases, telling you where electrons tend to accumulate, which gives you the excess negative charge, and where electrons tend to be withdrawn from, which gives you the excess positive charge. Now, if you imagine taking one of these bases and laying it down on another base, then you could imagine stacking the bases so that the positive region of one base, when the base is flipped over, interacts with the negative region of another base. And the negative region of the same first base may interact favorably with the positive region of the base that it's stacked on. And in fact, the geometry or angle between bases as you move up the stack of bases in double helical DNA or RNA reflects an optimization of the electrostatic complementarity, which leads to a favorable attraction between bases. And that's what underlies the base stacking energy. Now, why is it that hydrogen bonds are less important in the aggregate than base stacking in determining the energetics of double-stranded DNA, which is what the first question asks? To understand, we have to always remember that biological interactions are occurring in water. And water is itself a very strong hydrogen bonding molecule because it's highly polarized. And so the structure of water with the OH, H, with the delta minus on the O and the delta plus on the hydrogens is such that the hydrogens are extremely good H-bond donors, or the oxygen is the donor through the hydrogen, but the lone pair of electrons and the excess charge on the oxygen, excess negative charge, makes the oxygen an extremely good hydrogen bond acceptor. So when you think of the bases in DNA before they form the double helix, they are all hydrogen bonded with water. So their hydrogen bonding capacity is satisfied. So each of the bases has multiple hydrogen bonding potential for both donor and acceptor, and these hydrogen bonding donor and acceptor capacities are satisfied by water. Now imagine that the helices form, and so now you get the hydrogen bonding between bases, and now what's happened is that each base in the double helix has replaced some of its hydrogen bonds, originally with water, with hydrogen bonds with the other base in the double helix. So if you think of the energetic balance between the structure on the left-hand side and the structure on the right-hand side, in terms of hydrogen bonds, the energetic balance can be very close to zero. That is, the energy on the right in terms of hydrogen bonding is very close, very similar to the hydrogen bonding energy on the left. You've just replaced one set of hydrogen bonds for the other. And that is why hydrogen bonding alone would not drive the system to go from single-stranded DNA to double-stranded DNA. On the other hand, the base stacking, which is the interaction in the vertical direction, is unique to the double-stranded form. So base stacking occurs only in the double helix, not in the single-stranded form, at least as drawn here. And so the net effect, when considering the two terms, is that base stacking ends up favoring the double helix, and hydrogen bonding is a neutral player to a first approximation. However, it doesn't mean that hydrogen bonding is not important. Hydrogen bonds across spaces must be satisfied. Otherwise, if they were not satisfied, then a base would lose a hydrogen bond that it made with water on the right-hand side if it's paired against the improper base when it forms the double helix. So if the hydrogen bonds are not satisfied, otherwise, that is, if the hydrogen bonds are not satisfied, there is a huge energetic penalty. So hydrogen bonds are important in this way of thinking of things and making sure that the base that comes in to form the double helix is correctly paired in terms of the Watson-Crick rules. Because if an A is paired with a G instead of a T, then the hydrogen bonds in some instances will not be formed correctly and the bases would have lost hydrogen bond interaction with water, and that will be an energetic penalty. So the reason base stacking is important is because it's unique to the double helical form. The hydrogen bonds are seen in both forms, but they must be formed, and they ensure fidelity.